Hello, I'm Huanhuan Chen. In 2015, Arnold Beckman, postdoc fellow in Huanhuan's lab at Cornell Medicine. My research funded by the fellowship is focused on using stem cells to study cancer disease. I'm particularly interested in modeling small cell lung cancer using the cell resource derived from human embryonic stem cells and employing the model system for mechanistic study of this highly lethal disease.
Firstly, I will briefly introduce stem cells, my favorite cell type. What is stem cell? Stem cells are the cells that do not have specific functions, but they can replicate themselves, called self-renewal, and have the potential to become all other kinds of cells in human body. There are multiple types of stem cells in human body with different features. Among them, the embryonic stem cell hold the totipotency or pluripotency, which means the capacity to become all kinds of other cells in human body. Theoretically, each organ or tissue have a multipotent adult stem cells that can give rise to other type of cells in that organ or tissue. Because of the amazing properties of stem cells, scientists proposed to use the stem cells for many medical or research purposes: damage organ repairs, personalized medicines, artificial tissue engineering. Currently, I'm interested in stem cell approaches to modeling and studying cancer disease. In 1970, my mentor, Dr. Mammers, studied how a normal cell becomes a cancer cell, and the model he used was chicken fibroblasts. He first grows out fibroblast cells from chicken embryos and infected the fibroblasts with Rous Sakuma virus, the first oncovirus to have been described. The fibroblasts then transformed into cancerous cells in response to specific genetic mutations that were induced by Rous Sakuma virus. Nowadays, generation of human cells in different developmental stages through directed differentiation from human embryonic stem cell may provide us more rigorous human system to study tumorigenicity of normal cells. In my current lab, I grow the human embryonic stem cell in dishes and convert the cells into lung progenitor cells by manipulating cellular signaling pathway and growth factors. These lung progenitor cells can become functional lung cells, such as bronchial epithelial cells and alveoli type of cells. Of particularly importance, I developed a new method to effectively generate pulmonary neuroendocrine cells called peanut cell from human embryonic stem cell. Peanut cell is believed to be the predominant cellular origin of small cell lung cancer. With the availability of peanuts, it is possible to model small cell lung cancer in the right type of initiating cells. Then, through the inhibition of two genes called p53 and Rb, these two genes commonly inactivate in small cell lung cancer, I'm able to turn the normal peanut cells into cancer cells. As the results indicate, the peanut cell with p53 and Rb lost grow at a high proliferation rate in culture dishes and develop tumors when these cells were injected into mice. A major challenge in lung cancer biology is the identification of the cellular origin of different types of lung cancers. This procedure promises to provide a powerful context in which to explore the initiating and progression of small cell lung cancer in human cells by manipulating a small number of the genes. These findings indicate the utility of this system for studying the initiating, progression, and treatment of small cell lung cancer in much greater depth than has been possible before, using human cells and the gene known to be altered in advanced human small cell lung cancer. Our development of measurable experimental systems for studying the origin of small cell lung cancer using human cells that can be characterized at the single cell level and adapted to ask a wide range of questions about the origins and the control of this devastating disease. Many thanks to my mentors, Dr. Huanhuan at Cornell Medicine, and my great colleagues. I also want to thank my collaborators, Dr. Hans Schnicker's lab at Columbia University and Olivia Armento's lab at Cornell Medicine. Without their helps and supports, I couldn't finish this project. Last but not least, especially thanks to Arnold Beckman Foundation support, which enables me to successfully accomplish this project. This project as a whole will establish the foundation for my long-term research goals, to applying new approaches, to pursuing long-standing questions in cancer research. Thank you.